When 31-year-old Rachel Lawson was found unconscious behind a rural warehouse outside Buffalo, New York, detectives were stuck. No witnesses, no camera footage, no obvious suspect. The only clues were a torn piece of fabric clenched in her hand, a faint blood smear on her jacket, and a single drop of saliva on her wrist. To the public, the case looked hopeless. But inside the U.S. crime lab, this was more than evidence. It was a roadmap to the truth. Before we delve and hit the subscribe button, when the samples arrived at the forensic lab, technicians did one thing first: secure them with a strict chain of custody record. Every handler, every timestamp, every location. One mistake could make evidence inadmissible in court. In Rachel's case, the torn fabric, blood smear, and saliva swab were each sealed in sterile packaging, labeled, logged, and transferred to a controlled evidence room. This protects the case from contamination and manipulation. If you've never seen a DNA clean lab, imagine a hospital operating room. Then make it even stricter. Technicians wear full-body suits, double gloves, face shields, hair coverings. The air is filtered constantly. No outside DNA is allowed to enter. When Rachel fought back, she unknowingly grabbed her attacker's shirt. The tiny fibers under her nails held microscopic genetic clues. In the clean lab, technicians clipped fibers from her fingernails, isolated the blood trace, swabbed the saliva, separated them into DNA-safe tubes. Every movement is slow, deliberate, and recorded. Each sample could make or break the case. DNA is trapped inside cells. To release it, the lab uses chemicals and heat in a process called extraction. When Rachel's samples were processed, results showed: saliva sample, strong DNA profile; blood smear, low but usable; fabric fiber tray skin cells but degraded. This raised a chilling question. Was the attacker kneeling over her, holding her down, trying to move her? Every fragment hinted at a darker story. Before analysis, the lab must confirm one thing: how much usable DNA is present. A machine called a qPCR analyzer measured Rachel's samples. The saliva had more than enough, but the skin cells from the fabric? Barely any. Still, even one cell can change everything, if amplified correctly. PCR, the same technology used for COVID tests, copies DNA millions of times. This took Rachel's tiny samples and amplified them into full profiles strong enough to analyze. The blood and saliva belonged to two different people. One profile was male, one was female. Detectives realized Rachel may not have been attacked by one person, but two. The lab used STR profiling, examining the parts of DNA that vary between individuals. The results were shocking. The female DNA matched Rachel, confirming her blood was present. The male DNA from his saliva formed a complete, clear genetic fingerprint. The degraded fiber sample gave a partial profile, but the markers that did appear matched the male DNA. Both samples pointed to the same man. The attacker had gotten close, too close. The complete DNA profile was uploaded into CODIS, the National FBI DNA database. When the system scanned through millions of profiles, one name flashed on the screen: Ethan Ward, a warehouse worker fired 2 months earlier for harassment. He had a prior assault case, a sealed juvenile record, and he lived only 12 minutes from where Rachel was found. The evidence was overwhelming, but detectives needed a final confirmation. Police went to Ethan's home and collected a court-ordered cheek swab. When the lab compared it with the crime scene DNA, it was a 1 in 9 trillion match. Ethan cracked during interrogation. He admitted he followed Rachel after she rejected him at work weeks prior. He didn't act alone. His sister helped him stage the scene to look like a robbery. Her DNA appeared in the degraded sample, and Rachel's testimony confirmed it. Both were arrested. Rachel survived her injuries, and the case became a training model for how DNA processing saves lives. Please subscribe to our channel for more true crime content.